Good morning, everybody. We have come to the end of the excretory system of the renals, wherein you came to know about the functions of the kidney, wherein the cortical nephrons were concerned with excretion and the extramedullary nephrons were concerned with concentrating ability of kidney. And the major function of the kidney was to maintain the acid-base balance, which is called as acidification of urine. In today's class, you will know what is hydrogen ion secretion, how is bicarbonate reabsorbed, fate of hydrogen, the major buffer system, the bicarbonate, Dibasic and Ammonia, the reaction of urine with titratable acidity, which is based on the limiting pH of 4.4. So, cell function is very sensitive to changes in hydrogen ion concentration. The intercellular extracellular pH is maintained at very constant level between 7.34 to 3.5 to 7.4. Body pH is stabilized by the buffering capacity of the body fluids. So you can see what is acidic if the pH decreases below 7, and if it is more than 7 it is alkaline. And you can understand that in venous and arterial blood there is a variation. So, the average is between 7.35 to 7.45. That is the normal body pH. So, hydrogen is secreted normally from the cells of the proximal convoluted tubule and distal convoluted tubule. Acidification also occurs in the collecting ducts. This reaction is mainly based on the sodium hydrogen exchanger, which is based on the secondary active transport present in proximal convoluted tubule. Hydrogen ions formed by the tubular cells are in the presence of carbonic anhydrase, and the reaction is as follows. Carbon dioxide combines with water to form carbonic acid, which is dissociated in the presence of carbonic anhydrase to hydrogen and bicarbonate. Hydrogen ion from dissociation of carbonic acid is secreted into the tubular lumen. Bicarbonate diffuses back into the interstitial fluid. Sodium enters the cells from the lumen, which is coupled with the extrusion of hydrogen. So it is a sodium hydrogen exchanger. Conservation of bicarbonate is permitted by hydrogen ion secretion. So this is how the whole reaction in the tubular lumen, it happens in the cell, how water combines with carbon dioxide due to the cellular metabolism reactions, forms carbonic acid, which dissociates into hydrogen and bicarbonate, and it is exchanged for sodium. And the remaining bicarbonate is reabsorbed and the hydrogen ion is filtered. Transport activities, how it happens in the mainly because of the sodium hydrogen exchanger, which is a secondary active transport. In DCT and collecting tubules, hydrogen secretion relatively is dependent on sodium. It is secreted by ATP driven proton pump. Aldosterone also increases hydrogen ion secretion. The eye cells in the collecting tubule secrete acid. Some hydrogen is also secreted by hydrogen potassium ATPase pump. So hydrogen ion secretion and the acidification of urine occurs by two routes. The central theme of the exchange is in the cytoplasm for sodium ions in the tubular fluid, and reabsorption of bicarbonate is generated in the process. So the hydrogen ion is secreted. So this is how the sodium hydrogen exchanger and the total processes which happens in the whole nephron. As you all know, the acid-base balance is mainly controlled by the respiratory system and the renal system. To understand the concept of metabolic acidosis, respiratory alkalosis, respiratory acidosis and metabolic alkalosis, we need to understand how the renal buffers tend to perform. So for understanding, we need to know how the bicarbonate reabsorption takes place. 90% of the filtered bicarbonate is reabsorbed into the PCD by secondary active transport in the presence of sodium hydrogen exchanger. The bicarb, which is freely filtered, the secreted hydrogen reacts with the bicarbonate to form carbon dioxide and water. Both carbon dioxide and water diffuses back to PCT. The bicarbonic acid, which dissociates into hydrogen and bicarbonate, is forming the newly synthesized bicarbonate. So we need to understand where does the hydrogen ion come from. That is the sources of hydrogen and acid removal. It can be either volatile or non-volatile. It's normally only the 80 milliequivalents of the non-volatile acids are removed by the kidney. So body produces large amount of acid predominantly. Carbonic acid is volatile, which is removed by the respiratory system, whereas the non-carbonic, which is fixed, are the sulfuric acid, phosphoric acid, and HCl, hydrochloric acid. The three processes in which acid is removed are either by combination of hydrogen with the buffer, a reduction of carbonic acid, and reduction of non-carbonic acid by renal elimination of hydrogen. All body fluids are electrically neutral, is maintained by excretion of anions. Kidney too is important, so it cations to cover these, and hydrogen and ammonia is formed. Major sites of acidification in the kidney are distal convoluted tubule and collecting tubule. The fate of hydrogen in the urine is the amount secreted depends on the amount of acid secreted depends on the events in the tubular urine. It has a maximal gradient, that is, it is the limiting pH. If there were no buffers that tied up hydrogen in the urine, this pH would have reached rapidly and hydrogen secretion would stop. But three important reactions in the tubular fluid remove free hydrogen, permitting more acid to be secreted by the buffer systems. So what is a buffer? Addition of 3H to buffered solution results in one free hydrogen to be formed. The major buffer systems in the kidney are the bicarbonate system, the Dibasic phosphate system, and the ammonia system. So the bicarbonate system, wherein the newly synthesized bicarbonate is formed with every reabsorbed bicarbonate and a hydrogen is secreted. The concentration of bicarbonate in plasma glomerular filtrate is around 25 milliequivalents per liter. The secretion of hydrogen ion reacts with in the PCT with bicarbonate to form carbonic acid used for bicarbonate reabsorption. Carbon dioxide formed in the lumen returns to the tubular cells to form another hydrogen, so no net secretion occurs. Excess hydrogen ion is buffered by the remaining buffer system. When bicarbonate gets exhausted by the bicarbonate system, the phosphate buffer mechanism takes over. It has a pH which is identical. So this is Dibasic, where the hydrogen is secreted, combines with the monosodium hydrogen phosphate to form disodium hydrogen phosphate. So this mechanism is whenever there is renal contribution of new carbonate to plasma is achieved by tubular secretion, the intercellular bicarbonate and hydrogen and bicarbonate generation causes hydrogen movement into the lumen, so thereby bicarbonate into the plasma. In the lumen, bicarbonate combines with filtered phosphate, HPO4, and it is excreted as phosphoric acid. The secreted hydrogen reacts with alkaline phosphate to form acid phosphate, where disodium hydrogen phosphate combines with hydrogen to form Dibasic phosphate. Exchange of sodium hydrogen converts dibasic phosphate to acidic sodium dihydrogen phosphate, and this is excreted as titrable acid. The pH control is mainly by in the blood hydrogen exchange from the cells, and that's how the dihydrogen phosphate to monohydrogen phosphate. So disodium phosphate, that is alkaline, is converted to acidic phosphate. So once the bicarbonate and the phosphate system are utilized, the ammonia system, which is the third buffer in the kidney, comes into play. This is how the ammonia is formed. Ammonia is formed from glutamine, and it forms glutamate and ammonia. The hydrogen secreted from the cell into the tubular lumen combines with ammonia to form ammonium. Ammonia is diffusible, whereas ammonium is not diffusible. This is non-ionic diffusion. The reaction with ammonia occurs in PCT and distal convoluted tubule. Ammonia from tubular cells is mainly formed from deamination of amino acids, that is the glutamine. Glutamine is converted to glutamic acid and ammonia. The remaining 30% formation occurs by deamination of glutamic acid. The glutamic acid is converted to alpha keto glutaric acid and ammonia. The final 10% is by deamination of other amino acids and directly from the blood. The ammonia produced passes easily to the lumen by non-ionic diffusion. There it combines with hydrogen to form ammonium. Where ammonium is impermeable, it remains in the tubular fluid in the form of ammonia salts. Ammonia mechanism helps in sodium and bicarbonate conservation. So the reaction of urine and weak organic acids, the ultimate reaction depends on the hydrogen and acidic substance like dihydrogen sodium phosphate, that is acidic phosphate. Weak organic acids like lactic acid, beta hydroxybutyric acid, acetoacetic acid pass to tubular fluid by non-ionic diffusion. If ionized in tubular fluid, it remains there, and this accounts for titrable acidity of urine. In the DCT, the anions, which is mainly due to the lumen being negative, help secretion of more hydrogen ions. So let's come across what is titrable acidity. Each hydrogen ion that reacts with buffers contribute to titrable acidity. The newly synthesized bicarbonate accompanied by equal acid secretion in urine in the form of titrable acid, ammonia or both. It is measured by determining amount of alkali that must be absorbed to urine to return its pH to 7.4. It is largely attributed by phosphate mechanism. The factors which affect acid secretion are the intracellular PCO2. Higher the PCO2, more H2 bicarbonate is formed to buffer the hydrogen ions, and acid secretion is increased. Increase in potassium concentration, that is, when it is depleted, there is increased acid, or when there is excess of ammonia, acid secretion is decreased. And the enzyme carbonic anhydrase, when it is inhibited, it causes decreased acid formation. Aldosterone and other steroid concentration increases tubular reabsorption of sodium, which in turn enhances increased secretion of hydrogen and potassium. Reabsorption and secretion along the collecting tubule, that is mainly when water and solute loss is regulated by aldosterone and ADH. Reabsorption of sodium bicarbonate and urea is reabsorbed. pH is controlled by secretion of hydrogen and bicarbonate ions. So this is how the whole mechanism of bicarbonate, diphasic phosphate system and the ammonia system are involved in. The first is usually this is a general idea about how the GFR is filtered, and most of the solutes, anions, and cations are reabsorbed in the proximal convoluted tubule, and there is an increased hyperosmolality in the tube, in the medullary interstitium, due to the descending and ascending lumen permeability, differential permeability, and finally in the ascending limb and the collecting tubule, how ADH regulates the permeability.